Start recording. So it's the 29th of September 2017. We've been talking about bacterial cells so that we can figure out, and we've been talking about their DNA and what their DNA does and how it mutates so that we can figure out how we can have a bacteria pop up that's resistant. We're going to do we're going to take a sharp left turn for a minute. We're going to talk about a slightly different kind of cell because the process is easier to explain in this kind of cell, and then we're going to apply what we've talked about back to the bacterial cells. This is not a bacterial cell. How do we know? This is not a bacterial cell. Talk about it at your table. Tell somebody at your table how you know that this is not a bacterial cell, and Moser told me doesn't count. How do you know? What does it have that a bacterial cell doesn't? What do you think, Annalise? Oh, it's got a nucleus, and I heard somebody over here say nucleus as well. So this has got a nucleus. It also does not have a what around it? Cell wall. Okay. So you know what kind of cell this is? Eukaryote. So we said bacteria. That's pretty good for a first shot. We said bacteria are prokaryotes. You and 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 you are eukaryotes. Don't get excited. So is a maple tree. So is a ladybug. So is an earthworm. So is a cockroach. Ew. So is a dandelion. They're all eukaryotes. This one we happen to know is not a plant cell because plant cells also have a cell wall like a prokaryote does. This one's got to be some kind of animal cell. So it could be your cell. It could be a cell from an earthworm. It could be a cell from a mouse. It could be a cell from a cat, a dog, a blue whale, a lightning bug. That's a nice one. It's some kind of animal cell, but we could say it's your cell. So it's got, we're showing this membrane around it. There's a nucleus, and the nucleus has got little holes in it. It's got pores. What is a pore? Who here has seen a pore? Raise your hand if you've seen a pore. Okay, for those of you who are not raising your hands, I would like you to look very closely at your arm. Very closely. Do you see little holes in you? Do you see little holes? Are there little holes in you? Ah! You've been perforated! Ouchie. You've got pores. You've got holes in your skin. That allows materials. What's a material that comes out of your pores? Oil, sweat. It's a little hole that allows stuff to pass through. The nucleus of a cell has pores. They're actually called nuclear pores. Go figure. Really creative, folks. Very creative. So we've got this eukaryotic cell. What's in the nucleus? What's contained in the nucleus? Anybody have cell stuff in middle school? Does Mr. G do some cell stuff? Yeah. Okay. What's in the nucleus? You remember? What's in the nucleus? We got DNA and it lives in the nucleus. We've got this lovely, and this is my sort of sketchily drawn DNA. It's not pretty, but it'll do. I didn't bother drawing letters. I just drew little colors to represent the what pairs? Base pairs. Base pairs. So those little colored bars just represent the base pairs. And all of that DNA is down in the nucleus. Now, DNA is a crazy long molecule. We actually call it a macromolecule. What does macro mean? No, the opposite. That's what everybody gets wrong the first time. Micro means tiny. Macro means big. Big. Yeah, it's a macromolecule. It's huge. It's cumbersome. Remember the big, giant, messy pile of string that we had? This long, stringy, sort of awkward molecule. And we can, take this whole big, giant, long, awkward molecule, and I'm going to shrink it down. And I'm going to shove it down. Whoops, I just deleted it. That's not good. Hold on. DNA is a macromolecule, large molecule. There. You really can hardly see it, but I put my little tiny piece of DNA way down there into the nucleus. Okay, good. That's where it belongs. It lives in the nucleus. The DNA, in essence, When we say lives, obviously, it's just a molecule, but that's where the DNA is kept. You may have a particular place you keep things. Maybe you keep your cell phone charger in a particular place at home. As long as it's in that place, you know where it's at. Okay, good. Okay, Moser, where does this get us? What is DNA used for? Talk at your table. What is DNA used for? I heard it. It is the blueprint or the recipe for building proteins. You were so right. What do those proteins do? I don't know. I like to eat them. I'm going to have a hard-boiled egg at lunch. No, what does protein do? What do you think? Well, your muscles are made largely of protein. Anybody here ever get cut on anything? Are you still bleeding? Right now, okay. So, when you get cut, what happens? For most individuals, it does what? It clots. Yeah, it heals up, it clots. Your blood clots. Your blood clots because of particular proteins that exist in your blood. There's actually a whole sequence of ingredients that go into forming a blood clot. And it's this protein that has to be formed the right way so that your blood clots. Are there some people whose blood does not clot correctly? Yes, they're called hemophiliacs. Multiple variants of hemophilia, but it involves the misproduction of a protein. Oh wait, what was the blueprint for making proteins? DNA. We'll get to that when we talk about heredity. Okay, so the DNA is a blueprint for making proteins. Now where does your body make proteins? Proteins do a lot of things in your body. They make up your body. They make your eyes blue, they make your hair curly or straight, they make your blood clot, they make your muscles move. That's all a function of proteins. Where are proteins made? And you may remember this from cell stuff you did in middle school. In the Pac-Man part? Ribosomes! Yay! Okay, it looks like Pac-Man. Yes, it does, often. Proteins are made at the ribosomes. Where are the ribosomes? They're in this picture. Remember, we're only looking at a corner of the cell now. There's one, and 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 there's a big one, and there's one. You get the picture? All those little tiny dots out in the what of the cell? The juicy jelly stuff. What's the juicy jelly stuff called? The cytoplasm. So all those ribosomes, those little tiny dots that I've shown, out in the cytoplasm of the cell, that's where protein gets built. Awesome! Okay, so we know where the DNA is kept. We know where the recipe is kept, the blueprint. We know where the proteins are built. I got a problem. I got a huge problem. I like to cook. Okay, that's not a problem, Moser. You're off topic again. Back on topic. So I like to cook, and I've ended up at this point in my life, I use a lot of internet recipes because, gee, it's more convenient than climbing up and getting the cookbook down. And so I don't like to take my computer in the kitchen. It kind of lives out in the living room. And so I pull up the recipe on my computer, and I read it, and then I run in the kitchen, and I measure a few things. And then I forget what the third ingredient was, and I go back. And okay, I got it. And I go back in the kitchen. Because that's where I'm making stuff, is in the kitchen, but the recipe is out in the living room. This seems to be a problem for me. And my computer at home, I can't move. It's a big old clunky thing. I can't grab it and bring it into the kitchen like I could an iPad. Anybody see a potential solution to this? What, Gavin? I could write myself a note. I could write myself a note. I could jot down the recipe, and then I could go in the kitchen where I'm going to actually make whatever, the flourless chocolate cake, the spice cake. And then I would actually have the recipe right there, and I wouldn't have to run back and forth, and all the ingredients are there. And I have the recipe where it's being done with the ingredients. So here's my problem. Where's the DNA at? In the nucleus. I'll highlight that in red. Where are the proteins being made? Out here. Okay, great. So we just take the DNA out to the ribosomes, right? Do you know how much DNA is in your cells? A ton. Well, there are a certain number of individual pieces of DNA in every cell in your body. This many pieces of DNA. There's an actual number. There are 46. Yeah, it's a multiple of 23. So, you know, all of this is one piece of DNA, and there are 46 pieces like this in every cell in your body. How are we going to get this? So, well, we got those nuclear pores, right? Okay, great. Here's my nucleus. I've got a nucleus. That's good. I have a nucleus, and it's nicely sealed up to keep the DNA contained. There's a nuclear pore. Can you get the DNA out of that? Crap. This is a problem. Fortunately, Gavin's smarter than this. We can jot down the recipe that we need at the time. So even if I'm using a cookbook, do I need the entire big giant book? No, I need one recipe. Well, this entire long, I'm going to copy, oh, can I do it? Can I do it? I'm going to copy this DNA strand. This entire long strand of DNA, I'm going to momentarily hide, well, I'll just do this. This long strand of DNA codes for a whole bunch of things, codes for a whole bunch of proteins. Can tell your body how to do everything. In this DNA strand, I'm going to try to make the point that it extends even further than this page. It's huge. So we may only need a little chunk of it. You know what the little chunks of DNA are called? No, we're getting there. Little individual pieces are called genes. You knew that. So a gene is just a short section of DNA. So maybe we only need one gene, because we're only building one recipe. We're only building one protein at a time. So how do we go from these 46 giant long pieces of DNA to the one little chunk that we need to get it out to the ribosome? Well, I'm glad you asked. I'm going to get rid of some of this. So now we're zoomed down to the nucleus, and we've got that piece of DNA. And we only need to build one protein from one little section, one little gene. So what's the first step if we want to well, we talked about replicating DNA, but now we're going to make a copy of just a part of it. Would we still have to unzip it? Yes. So just like in synthesis, guess what the first step in building a protein is? We have to unzip the darn thing. We have to unzip the darn thing. And I'm doing a really ugly job drawing this. But it has to get opened up, and then we write a little note. We just sketch down the part of the recipe that we need. That gets made on a little copy. And oops. And then that little tiny copy, and you may have noticed something weird there. That little teeny tiny copy is going to actually go out through a nuclear pore and out to a ribosome where we can make proteins. So we have to take a copy of the section of the DNA, write down the recipe, pull it out of the nucleus, and go out to where the proteins are being built. Now we're going to look a little bit more closely at that process. Well, actually, no. You know what? We're not. For today, we're going to stop there. And I'm going to have you work on some of the vocab, picking up terms that you feel more comfortable on. Here's one thing I want you to do. Remember what I talked about with the social brain? We said that your brain is hardwired to sort of get high from social interaction. That's why Facebook and all the social media stuff is so addictive to humans, because humans have gotten rewards over a couple 100,000 years from working together with other humans, so we like it. So, on that part C, for any word that we discussed today, because if you're talking with other people about it, different parts of your brain are lighting up, and you're getting more bang for your buck, brain-wise. So I want you to look at those part C's, discuss them together. I had some people who turned them in last time and said on part C, not really sure. You can't say that. If you're not sure, ask. Talk to somebody else. If nobody at your table gets it, maybe we haven't talked about it yet, or maybe we haven't talked about it well enough yet. So because we haven't gotten any further than this in the protein synthesis discussion today, I'm going to have you, I'm going to borrow yours for a second for a prop. If you still have this sheet from Amoeba Sisters, I would like you to go ahead and turn it in. I know you have 3 and 4 undone. I'll check them off and give them back to you, because I think we're going to start to do the protein synthesis for real on Monday. Okay, so I want you to take this time to be working on the part C vocabulary. Yes, ma'am. Yes, that's due today also. Both of the video sheets are due today. Questions? Comments? Concerns? Okay, work.